Hi students, so today I want to go over the sliding filament theory on how skeletal muscles contract. This tends to be a difficult topic. There's usually areas of confusion on how this works, so I thought this would be a good lesson to go over with everyone. So we're going to start with a basic review of the skeletal muscle. First of all, the skeletal muscle is one of three muscle types. There's the smooth muscle and the cardiac muscle as well. But the skeletal muscle is unique in that it is going to be highly structurally divided. It's going to have very specific parts to it. And it also is one of the few cells in the body that is multinucleic, which means that there's multiple nucleus throughout the cell, and that's to help with controlling of the massive length of these cells for contractions. The nuclei, which is just plural for nucleus, will sit just beneath the cell membrane, and in a muscle cell, we call the cell membrane a sarcolemma. So those nuclei sit just beneath that cell membrane. Okay. Within the muscle cell, one of the organelles, and this is not something you cover in biology class, most of the time in biology, you talk mostly just about the nucleus, mitochondria, endoplasmic reticulum, those basic overall types of organelles that most cells have. But muscle cells have a lot of organelles called myofibrils. And these are these little skinny cylinders that you can see pulled out on this picture right here. And they're stuffed inside and they're filled with proteins. And these proteins are what are going to contract and cause the muscle cell to contract, which when a bunch of those cells contract, the entire muscle will end up contracting. So with the sliding filament theory, when we talk about that, we're actually talking about contraction on the level of these little myofibrils where these protein fibers are going to be found. So on that microscopic level is what we're talking about, but that's the basis for the entire contraction of the skeletal muscle as a whole. So the myofibril is, like I said, an organelle that is very specialized with proteins inside of it that are going to contract. These proteins are called myofilaments. Notice the prefix myo, myofibril, myofilament. Myo just refers to muscle. Myo is a prefix that determines muscle or distinguishes muscle. So a myofilament is just a protein, and there are two main proteins that are found in these myofibrils, the actin and the myosin. I like to remember the difference between the two in that the actin thin, so actins are the smaller, thinner ones. When I say smaller, I mean thin, and myosin is the thicker one. Myosin has the big O in it, and O's are big and fat, so myosins are going to be our larger, thicker proteins that are in these myofibrils. And the way that these myofibrils align themselves within the cell is going to give that banding or striping pattern that we call striations. Or if you remember from the skeletal muscle tissue as well as cardiac muscle tissue, they are the two types of muscle tissue that have these striations or striping methods to them, and that is just based on the overlapping of these different proteins within their cells. So when we get inside of the myofibril, inside that organelle, and look at how the actin and the myosin layer on top of one another, and these proteins layer on top of one another within those little organelles, we can see that there's going to be the thick red myosin. Okay, so this big red band here in the center of this picture is what is the myosin protein. Notice it's big and thick. And then the thin protein fibers are these little blue, they look kind of like braided or twisted helixes, these little blue thin lines. Those are those are the actin. Actin thin, myosin thick. Okay, so the myosin's the big red one, the thin blue one is the actin. And they are going to be separated into groups called sarcomeres. The sarcomere is just considered the contractile unit of a muscle fiber. So they kind of just line up next to each other, side by side by side by side. It's kind of like bricks, laying bricks along each other. So each sarcomere sits next to each other. Each group of myosin and actin are going to be stacked next to each other, these sarcomeres. And we kind of divide this sarcomere, or this this grouping of myosin and actin into different zones. So the middle of a sarcomere, the middle of where the actin and myosin are going to overlap, is called the end line, M for middle. The ends of each sarcomere is called a Z-disc, kind of because it's like a zigzag little pattern here that kind of looks like a Z. So that represents where one sarcomere ends and where the next sarcomere is going to begin. So where one set of actin and myosin end and the next set of actin and myosin begin. Okay? And as we look at this, we can look at how these slide over top one another. So it's called the sliding filament theory because the proteins literally slide across one another. And as they do so, it pulls the ends of the sarcomere closer together, which causes a shortening of that cell. And when you have a bunch of cells shortening at one time, then that actually ends up being the whole muscle contracting, because contractions are shortening of the muscle. So in the sliding filament theory, you can see we have our sarcomere here at the top, and this is what a relaxed sarcomere looks like. We have a big center where it's just myosin, just the red in the center, and there's no actin all through here in the middle, and it's called the H zone. So in a relaxed muscle cell, the H zone is the area where there's just myosin in the middle and no actin. Then you have the A zone. The A zone includes that middle portion, but also includes where the myosin and the actin already overlap as well. So there's going to be portions on either end where their myosin is overlapping a little bit with the actin, and that is going to be the A zone, which is going to be that whole section from end to end, basically, with the middle included. And then, of course, you have your Z lines, which is where the ends of the sarcomere are. So what is happening here is on the myosin, these little bumps that you see along the edges of the top and the bottom are called myosin heads. They kind of look like golf clubs if you were to get really close up to them, kind of sticking out. And these golf clubs want to attach to the actin, the thin blue lines. They want to attach to it, but they can't in a relaxed cell. And they can't because the actin is protected. It's protected by proteins, troponin and tropomyosin, and those are going to be covering the binding sites while the muscle is relaxed. But when the muscle is stimulated by a nerve, it causes a release of calcium ions. And you see this coming in this bottom picture. Calcium ions start flowing into the cell, and these calcium ions attach to the troponin and the actin, causing the binding sites to release. So it actually moves that protective covering off of the actin and allows the binding sites to open up so that those little golf clubs that are on the red myosin can actually attach to the actin. And when it does that, they actually pull. They move kind of like a power stroke. If you think of like an oar going from forward to backward. And so it's actually going to pull on that actin. And when it pulls on the actin, you can see here that H zone disappears. The middle disappears. We no longer have any portion of the sarcomere that is just myosin. It's all myosin and actin overlapping because they've slid over top one another. And that has shortened our sarcomere. Okay, that has shortened the sarcomere. Now the A zone, if you notice, if you look closely, this A zone line matches up pretty much still with the A zone line in the relaxed muscle. The difference, though, is that the I zone, which is the space between one sarcomere end with myosin and the other sarcomere end with myosin, has now decreased and pretty much disappeared. So the spaces between those sarcomeres is no longer there. We have smaller spaces, so as all of the sarcomeres are kind of pulled towards one another, towards the middle, so that causes the shortening of the muscle. So it's all based on that myosin connecting to the actin, pulling on the actin towards the middle, which causes that shortening to happen of the sarcomere, which then shortens as a whole the muscle cell and eventually the muscle as a whole. Okay, and it's all because of the calcium ions coming in and releasing those binding sites on the actin so that the myosin can connect. Now also keep in mind that ATP, which is the energy that the cells use to do work, is also required for this to take place. So while this is happening, ATP is also being used to allow for this to take place. So that is why we are constantly needing to replenish our oxygen levels in our cells when we are exercising or doing work in order to allow for ATP to continuously be made so that the cells can do this work at an efficient manner. Okay? All right. So now that we kind of have a better idea of how the muscle contraction works on that microscopic cellular level, let's just talk a little bit about some rules, or I should say some guidelines that go along with the contractions of a muscle cell, or I'm sorry, of a skeletal muscle as a whole. So with muscle cells, and notice that on this first bullet here I said muscle fiber contraction is all or none. Keep in mind that the term fiber and cell are interchangeable. So when we say muscle fiber, we're talking about one individual muscle cell. So a muscle fiber contraction is all or none. What this means is if the muscle cell is stimulated at one end by a nerve, the entire cell is going to be stimulated. There's no partial stimulation. Not half of the cell can be stimulated and the other half not. It's all or nothing. Also, within the skeletal muscle, not all cells may be stimulated by a nerve at the same time. So this is why we sometimes get muscle twitches. Okay, when we get a muscle twitch, that's because some of the muscle cells are being stimulated and others are not. Also, this is how we variance the power or the force within our muscle movement. So if we're trying to lift up let's say we're trying to lift up our pencil to do some writing, we don't want to use the same force that we would use to lift a desk. And so that's based on the number of cells within the muscle being stimulated at that time. We want more cells involved when we have to lift a desk. Not as many have to be involved when we're lifting a pencil, otherwise we would probably jab ourselves in the eye. All right, so these different combinations of muscle cell contractions or stimulations offer or give us different responses and different forces. And so the force of your muscle, the amount of power we have when we are using our muscles, is dependent upon how many of the muscle cells are being stimulated at a time within that muscle group. All right, muscle tone. A lot of people talk about muscle tone, and especially if you exercise and if someone has good muscle tone and that type of thing. And this is based on the fact that there are always going to be some muscle cells contracted in your muscles at all times, even when your muscle is not really being forcefully active. So even if you're just sitting at your desk or laying in bed, your muscles in all your body have some cells that are contracting at all times, and that is what we call muscle tone. And that's just to help make sure that the muscle is not just this flabby thing that just lay there. It just gives the muscle the tone and is constantly having alternation. It is an involuntary thing. We don't think about the contractions that are taking place when we are at rest and that type of thing, but that's what is muscle tone. Muscle tone is the fact that you're going to have some cells in a muscle contracting at all times, even when you're relaxed or not actually physically using that muscle. All right, and then movement of muscles in the body. It's important to know that muscles only work because they have one fixed attachment and one moving attachment, and that attachment, the muscle crosses a joint. So that's how it makes the movement take place. Muscles have at least 2 attachment points. Sometimes they have 3 or 4, depending on the muscle. For instance, a tricep, tri means 3, so it actually has 3 points of attachment. So that's something to kind of keep in mind. The quadriceps as a whole are actually 4 different muscles, but they each have their own different attachments, so 4. But there's always going to be at least one origin, which is the point where the muscle is attached to the immovable bone, and then the insertion is the attachment to a movable bone, the bone that's going to move when it's contracting. So if we look at this picture here, we have the bicep, and on the bicep, one of the origins is actually on the humerus, which is the upper arm bone, and that's the immovable bone. So when we contract our bicep, the bicep pulls on the ulna here, is actually going to pull it up towards our upper arm and cause the contraction. So the movable attachment is called the insertion, and it's down on the lower arm bone, and that's what gets pulled to move up. Whereas the non-movable attachment for the bicep is up here on the origin, which just means that the muscle, the upper arm bone, is not going to do any movement when that bicep is being contracted. All right, there are a couple muscle rules to keep in mind. Muscles only pull, they do not push. So that's something to remember. When your muscles are contracting, they're only pulling on something. So for instance, in that bicep, let me go back to it. In this bicep one, we have a pulling taking place on that lower arm to bring it up. And when you go to straighten that arm back out, the bicep is not going to push back down. What's going to happen is that you're going to have a tricep, which is on the back side of the arm, and the tricep is going to then pull on the back side of the lower arm bone to pull it back down. So it's always a pulling motion. So that's something to remember. Muscles must always cross at least one joint. Joints are where the bones meet, and if they don't cross a joint, then there's no movement that could take place, because the joint is where the movement is allowed. So they have to cross at least one joint. The insertion will move towards the origin, so the point where the muscle attaches on the bone that's going to move is going to be the point that moves towards the non-movable attachment. And then another thing that's kind of neat to know is a little trivia thing that a lot of people don't think about. Muscle fibers will increase in size but not number. So the more you work out and you may get bigger muscles, you're not actually getting more muscle cells. You're just increasing the proteins that are within those cells, causing the cells to get fatter and bigger, which overall causes the muscle as a whole to become bigger. So muscle fibers increase in size, not number. So that's kind of an interesting thing to keep in mind. All right. So hopefully this helped clear up a little bit about the sliding filament theory in particular, because I know that's one that tends to be a little bit difficult and daunting when looking at it as a whole, and also just a quick review on some other basic information about overall skeletal muscles. And as a treat for watching and attending this lesson on the muscle sliding filament theory, I am going to share with you a assignment template that you can use for the 2.03 assignment, and this can be submitted in place of the 2.03 assignment that is described in the lesson. So let me open up this file here for you. 1 second. All right, and it's called 2.03 Muscle Contraction. It's going to be a PDF file. All right, and that should be complete now for you. So you should be able to see that, and you can actually now save it. Okay. Okay. All right. So I thank you guys for coming and joining me today. And if you, of course, ever have any questions, or if there's anything else that I did not cover, or maybe you still are not 100% sure of, please make sure you contact me, and I will definitely go over that with you. Have a wonderful day. Thank you.